Greetings, my name is Ramin Sarkar, and today I'll be talking to you all about the utilization and analysis of herbal treatments for Alzheimer's disease. Starting off with a little bit about myself, I'm a rising senior at the Gwinnett School of Mathematics, Science, and Technology, which is located in Lawrenceville, Georgia, right off of Atlanta, Georgia. I've been doing independent scientific research for about 2+ years and won an international award and a scholarship from ACS. Additionally, I'm an intern at the Mercer University College of Pharmacy and currently testing indole 3-acetic acid alongside a professor for treating vascular dysfunction and hypertension in spontaneous hypertensive rat models. Additionally, in the upcoming months, I'll be doing a mentorship with Emory and Mercer for a future research endeavor. And I'm going to be designing a research program for interested high school students at the Philadelphia College of Osteopathic Medicine and becoming a lead mentor there. And lastly, I am a current hospital volunteer at the Northeast Georgia Health System. And my current interests are golf, hiking, pickleball, and neuroscience. Starting off with the introduction and global problem of Alzheimer's disease, Alzheimer's disease is one of the most progressive neurodegenerative diseases an individual may possess. And this can lead to the impairment of cognitive function, behavioral function, memory, and even movement, overall globally affecting 55 million people worldwide. And as of today, there are no top definitive treatment options available for Alzheimer's disease. Although there are many on the market right now, they're not definite and not going to help treat the disease on many other people. And currently, 98% of Alzheimer's treatments have failed, with a only 2% success rate for most treatments. And therefore, natural herbal treatments are actually a popular medication source for Alzheimer's, and it has been proven that natural herbal treatments can actually lead to the treatment of successfully treating Alzheimer's disease. And the four current natural herbal treatment extracts that I researched were ginkgo biloba, lion's mane, basil, and sage herbal extracts. And this is because they contain the highest amounts of the anti-amyloidogenic, anti-inflammatory, and antioxidative properties according to the literature. In order to pursue this research project, I used a model system called SAN or Hepatitis elegans, also known as C. elegans, which is the nematode roundworm that is used for neurodegenerative disease research and genetics research. There is also a 60% homology between human homologous genes and C. elegans genes. Additionally, the proteome in the C. elegans consists of 83% of humans' CGI genes. And overall, my research goal starts off with the null hypothesis stating that the behavioral characteristics of C. elegans and the protein concentration, also known as the molecular content, will remain unaffected when exposed to the herbal extracts. However, in my experimental hypothesis, I state that the behavioral and characteristics and the molecular characteristics also is the protein concentration will be affected when exposed to the herbal extracts. And this is mainly due to the fact that there are many neuroprotective properties of the herbal extracts that may alter the C. elegans behavior and the molecular content. And moving on to my methods, here's a quick rundown. My independent variable is the type of herbal extract, and my dependent variable is the C. elegans behavior and molecular effects when they're fed the extracts. And the control variable are the C. elegans with no herbal extract treatment. My C. elegans behavior was defined by the C. elegans speed, response to external stimuli by tapping them, and their distance and their response time after tapping them, and the foraging behavior, which is the distance to locate for food and the time to locate for their food. And lastly, the C. elegans protein concentration. Going through a quick rundown on my lab protocol methods for the behavioral analysis, I first started by sorting off the C. elegans culture plates, where 4 is respective to 4 of the herbal extracts. And then I created their food source and the treatment together, where the food source is E. coli K12, and I mixed it in with 1 milliliter of the herbal extracts respectively. And then I incubated all the plates for 24 hours for 25 degrees Celsius and then transferred them respectfully. And then at the end, I applied autofluorescence adapters and filters to a stereo microscope and recorded them and inserted them for data collection on a worm lab application software, which is normally used for C. elegans behavioral analysis. Now, for the lab protocol methods for the molecular side, I used the basioconsinic protein assay, also known as the BCA protein assay, in which I gathered the cell lysates by centrifuging all the C. elegans to get the genetic material and rinsed them out to get the denatured proteins. Additionally, I created BSA standards in order to follow the process of the BCA protein assay and inserted and put the cell lysates and the denatured proteins into a well plate and put it into a well plate reader, also known as a spectrophotometer, and measured the absorbance and calculated for the standard curve to eventually get protein concentration. Now, here are some results of my whole analysis, and I found that ginkgo biloba was specialized out of 3 out of 5 behaviors, which is shown here where ginkgo biloba actually exerted highest speed out of all the other C. elegans. For the herbal extract treatments versus the H. elegans protein concentration, ginkgo biloba actually yielded a higher protein concentration compared to all of the other herbal extracts. Additionally, for the external stimuli responses, ginkgo biloba had a higher distance traveled and ginkgo biloba having a lower response time, meaning they were more faster. Additionally, for the foraging behavior, here the control actually benefited from the foraging behavior for both the analyses. And for my statistics, I used an ANOVA statistical analysis test to assess whether there were any significant variations in the behavior and protein attributed to the different herbal extracts. And I found that 5 out of 6 of the tests were shown to be statistically significant, showing that the null hypothesis was more rejected than expected. And overall in this discussion, I found that ginkgo biloba exerted the highest specialization of the C. elegans behavior, which was the C. elegans speed, response to external stimuli, and protein concentration. Here are my references, and I would like to acknowledge the GHLC members, my research teachers and my mentors for providing me constructive feedback, my family, and all of you all. And I would like to thank you to my wonderful audience. If you want to contact me for more information, or you have any questions about my research, here's my contact information below. Thank you very much. Have a good day.